Hey guys, Mr. McDonald here. Today we are going to talk about incomplete dominance and codominance. And this is kind of the next step from what we talked about last week. Last week we talked about complete dominance, where one gene or one allele totally dominated another one. And as we've learned more about genetics, we've found out that not all genes work that way. And there's these other instances of incomplete and codominance that are also out there. All right, so to kind of use a metaphor or a simile to kind of help you guys out here a little bit, in complete dominance, it's kind of like when you and your friend want to go to the movies, and you decide you want to go to the movies, and they decide they want to go get something to eat. All right, going to the movies, let's just say, is the dominant trait. So you don't go get something to eat, you only go to the movies, and that's all that's expressed in the phenotype. In incomplete dominance, there's a compromise. We meet somewhere in the middle. So maybe you guys go to the movies, and while at the movies, you get popcorn and a bucket of soda. And then that way, you guys get to eat as well as see the movie. In codominance, you do both. So that means you'd have to stop at Burger King on the way to the movies and, grab a chicken sandwich and a Whopper and then go to the movies. All right, so we're going to kind of walk through incomplete dominance and codominance in this video. So starting with incomplete dominance, we see that we have to talk about hybrids. And hybrids, again, are going to have one dominant and one recessive allele. All right, so let's go through an example here. We're going to use flowers because this is pretty common in flowers. They use snapdragons. It's common in petunia, pansies, roses, you name it. They all have incomplete dominance. All right, so in the case of homozygous dominant or homozygous recessive, our two capital R's, that individual would have red flowers, while the two small r's is going to have white flowers. Now what we're looking at is if we cross those two flowers, what are their offspring going to look like? So let's check that out. All right, so again, there's one individual in the cross, there's the second individual. So let's make a Punnett square. All right, so there's the red flower on the left-hand side, the white flower on top. Come on, here we go. And then there is our potential genotypic possibilities of the next generation. And as you guys can see, we only have actually one genotype, all four are the same, and it's capital R, small r. And in this case, a red parent and a white parent produce a pink parent. Or I'm sorry, a pink offspring. So that's different than in complete dominance. So here, again, we meet somewhere in the middle. We have incomplete dominance going on here. All right, so our, again, first generation here of offspring are all going to be pink. They're going to be heterozygous. All right, so we're meeting somewhere in the middle, and that's the idea of incomplete dominance. It's a compromise between these two different genes. This gene can't totally outcompete this gene like they do in complete dominance like we worked on last week. All right, so if we check out this picture, again, you can see we have that red individual with a genotype of capital R, as well as a white individual with a genotype of small r. And in the first generation of offspring, they are all pink, just like we saw in our Punnett square. So these guys all have a genotype of capital R, small r. We would say that they are heterozygous or hybrids, and that's our first generation. Now that doesn't mean that the red and the white individuals have been completely removed from the population. So let's go a step further here and move on to a second generation. Okay, so this is like your grandparents' generation, your parents' generation, then finally you. Okay, so in this generation, which, for the sake of our example, that would be our parents' generation, these guys ignore the fact that their brother and sister are going to cross, and they're going to get us these possibilities. Homozygous recessive, 25% shot. Homozygous dominant, 25% shot. And we have a 50% chance at being a hybrid or heterozygous. If we cross this flower with a white flower, we have a 50-50 shot. Half them will be white, half them will be pink. So the point I want you to take from this is that the original white and red phenotypes do not disappear from the population. They're still there, and they can still be expressed in future generations. Okay, so again, that's incomplete dominance. So now moving on, we're going to check out codominance. Again, going back to our movie theater, going out to eat example that we started with. In codominance, you have to do both. So you need to stop somewhere to eat on the way to the movie theater. All right, so places where you see this in everyday life. If you've ever seen a dog like a Dalmatian or a Jack Russell Terrier that has spots, those are examples of codominance. Horses, palomino, ponies, examples of codominance. Our blood, which we're going to look at here a little bit, is an example in the human body of codominance. And in codominance, again, both genes are expressed at the same time. There's no compromise. There's no domination of another one. Both of them are expressed. All right, so that's what it says here. 2 alleles are expressed in heterozygous individuals. All right, so again, let's check out blood type. So one type of blood that you can have in the body is type A blood. All right, so here we have IA and IA, so that would be homozygous dominant. All right, or it could also be expressed as IA in this small I. We'll talk a little bit more about the small I here in a minute. You can also have type B blood. So we have IB, or again, we can have this weird hybrid over here of IB and I. Or we can have type AB blood, which gets us IA or IB, and that produces type AB blood. All right, so a little bit about what this means. Type A blood means that in the cell membrane of the red blood cells, there is a little sugar attached to some of the proteins that sticks out, and that's type A sugar. Type B is a different type of sugar that sticks out again from the proteins embedded in that red blood cell, and that's type B sugar. And then we have this third type of cell that has both sugars. So type AB has the type A sugar embedded in its cell membrane as well as the type B sugar embedded in its cell membrane. All right, but there's yet a fourth type of blood cell out there, which is what I happen to be, type O. And type O has a genotype of small I. And type O red blood cells have no sugars embedded in their cell membranes. So why is this important? This is important because of blood transfusions. And back in World War II, 60, 70 years ago, we didn't know about the different types of blood. And we didn't know why blood transfusions worked while other blood transfusions didn't work. And it has to do with these different proteins and sugars that are embedded in our cell membranes. So if we check this out, a human with type A blood can't get a blood transfusion from someone with type B or type AB blood because of these B sugars. Their body would reject it. All right. Same thing with somebody with type B. They cannot get a blood transfusion from someone with type A blood or type AB blood. They can only get a blood transfusion from someone with type B blood or type O blood. Type O can give blood to anyone. They're known as universal donors. All right, where the type O individual can only get type O blood. All right, this individual can't get type AB blood, type B blood, or type A blood. It would reject all three of those. And again, that's because of these different sugars that, again, are embedded in their cell membranes. So that's just a little bit about blood types in humans. There's also the Rh factor, which is positive and negative, and we don't need to get into that here. That really doesn't deal with codominance. But anyway, that's a little bit of background about human blood. So let's check out a Punnett square here. So we have a homozygous male for type B blood, and there's the genotype in parentheses. And that individual is going to have a child with a woman who is heterozygous for type A blood. And again, there's the genotype in parentheses. So let's check out our Punnett square. All right, so what does this mean? Well, it means that we have two possible outcomes for their children. We have a 50% shot at type AB blood and a 50% shot at type B blood. All right, so medically speaking, beyond that, so that means that this individual would not be able to receive blood from his mother, okay, because they would be type B. All right, so let's check out another one. So another codominance problem. So we have a type O male and a female that's type AB. Again, the genotypes are in parentheses. Let's check out our Punnett square. We fill them in. We complete our Punnett square. And what we see again is 50-50 shot at type A blood or type B blood. All right, so again, the father could donate blood to either of the children. The children could not donate blood back to their father. But either child, or any way the children would shape out, they would be able to donate blood back to their mother. All right, so we have one last question here. Again, trying to drive home the idea of codominance. If a boy has a blood type O and a sister has a blood type AB, what are the genotypes and phenotypes of their parents? Okay, so in this case, they're giving us the middle of the Punnett square, and we have to fill in the outside of it. So let's check that out. So the boy's genotype, since he's type O, has to be small I, and the girl has to be IA, IB, because she's type AB blood. So let's go to our Punnett square. Looks like my head's in the way. I apologize for that. So that's what we know has to happen. And because we know that, we can fill in the other parts of our Punnett square. So our parents had to be type A and type B in order to get children that have those blood types. All right. So anyway, that's dominance and codominance. I assigned you guys 2 worksheets, one on dominance and one on codominance. They're found on Schoology. Those will be due on Schoology Friday night as opposed to Sunday night like they were last week. You guys can hand them in the same way you did last week. That worked out really well. I got those graded and got those turned around to you guys pretty quickly. And hopefully you guys are okay with that. Again, I know you've done Punnett squares in the past. So please check those out. If you have any questions, please let me know. Again, my office hours, 8 to 10, Monday, Wednesday, and Friday. You can shoot me an email anytime, and I'll get back to you eventually, and we can set up our own private time if you have a lot of questions where we can Zoom or Skype or do whatever it is you do to get you guys straightened out. Have a good night.